All right, so we were looking at gene expression. Okay, so this is where we have sequences of nucleotides in the DNA. So the nucleotides, the A's, T's, G's, and C's, are copied into RNA. So then we have RNA copy A, U, G, and C. The RNA is the single strand, the DNA is a double strand. It's not the whole DNA. We're only copying a section of the DNA, a piece that's called a gene. That RNA could be a particular type of RNA that becomes either messenger RNA, transfer RNA, or ribosomal RNA. And then what we have are all three of these types of RNA are going to work together in a process called translation. So instead of transcription, it's translation. So we're changing the language from nucleotides to amino acids. And we get a polypeptide. Right, so which will essentially be or become at some point a protein. So usually we say, or there's this older saying, very old saying, says one gene, one protein, and that's not really true. Sometimes it takes multiple genes to get a single functional protein. Sometimes one gene can be edited here at the RNA stage and result in multiple different proteins all from the same gene. But the process is still going to be essentially the same. DNA is copied into RNA. RNA is then working together to make the polypeptide that eventually, in some way, will become a protein. So what we're going to look at now is the translation process, the process where these three actually are going to work together. Right? This process is going to happen in the cytoplasm. Now that's for both prokaryotic and eukaryotic cells. Prokaryotic cells have no choice. There is no nucleus for this process to occur in. The transcription process happens in the nucleus of a eukaryotic cell, also, but in a prokaryotic, it's still going to be in the cytoplasm. So bacteria in the cytoplasm, and eukaryotes for the cytoplasm for this process here of translation. What we have, or we're going to start off with, the process is something called initiation. So with initiation, so we're going to have it here. Initiation is where we're going to get a transfer RNA carrying the amino acid methionine. So we call this a met tRNA. And so this is a transfer RNA. And it has the amino acid methionine attached to it. Here in the bottom loop, if you remember from looking at transfer RNA lecture, it has an anticodon. And this particular anticodon is going to be UAC, and we'll see why it is that in a little bit. Now, that's the first part. Methionine transfer RNA is then going to attach to the small ribosomal subunit. So this is the 18S ribosomal RNA, or the small subunit. And together, these two things, the transfer RNA with methionine and the small subunit, create something called the initiation complex. All right, so the initiation complex are these. Now we get to the next step. So what else do we have in this process? Well, we have now some transfer RNA here, and here's a little bit of the ribosomal RNA, not all of it. But the messenger RNA is what actually contains the code, the sequence of nucleotides. So that's what we're going to get to. I'm going to use a different color here. Yeah, next. So messenger RNA is more linear. If you remember at the 3' OH end, we're going to have a whole bunch of adenine nucleotides, the poly A tail. At the 5' end, where there would be a phosphate, you have a backward GTP added, which creates the 5' cap. So that's the 5' cap. Now, what's going to occur next, and this is still initiation, is this initiation complex is going to bind to the 5' cap. that's going to be part of the importance of that 5' cap. We said without a 5' cap, the messenger RNA is unreadable. So typically it isn't read. There is no translation process. The information isn't used to make any polypeptides or proteins. So we need this cap to be there. Also, if the tail falls off or the tail gets too short, it doesn't actually fall off, but enzymes are chopping up the tail, shortening it the whole time it's in the cytoplasm. And once the tail gets too short, it does initiate a signal for the cap to be removed, which then doesn't allow the initiation to even occur. So the cap is very important for the binding of this initiation complex. So they bind, and then what's going to happen is this initiation complex is going to start moving along the messenger RNA, and it's going to be looking for something. All right, what's it going to be looking for, right? So it has the anticodon UAC, so it's going to be looking for a particular codon, right? It's going to be looking for the codon AUG. So AUG is going to be what we call the start codon. And this particular transfer RNA has the anticodon to match it. So it will bind to the cap, it'll move along, and there could be, quite a number of nucleotides before the start codon. And it's not going to read them. It'll kind of look at them. They don't match up, they don't match up, they don't match up. And then eventually when it finds the start codon, then the next step is going to occur. And that is that the large subunit will come in. That's the one with the 28S, the 5.8S, and the 5S ribosomal RNA. So all the types of RNA are now together. We have transfer RNA with an amino acid, and the messenger RNA is being read. That contains the information. This is just initiation. This is how it all gets started. So all three types are now together. And that's pretty much the end of initiation, and we'll begin into the next phase called elongation. Okay, so after initiation, we have elongation. Elongation is somewhat of a then repetitive process, and I'm going to try my best to kind of animate it here, kind of showing you step by step, because it's an active process. There is change, there's movement. The ribosome is moving along the messenger RNA. It's going to end up reading the whole sequence. The sequence is, we don't know what it is right here. I'm just making up a sequence. Okay, so you can just see it's just a bunch of nucleotides, and together these nucleotides are going to spell out a code that's going to be used to make up the amino acid sequence of this protein. That code is broken up into groups of 3 nucleotides that are called codons. Okay. So now it's going to read one codon at a time. So that's one cluster or group of 3. So now we're going to move into the next codon. But what we have currently at the beginning of elongation is the transfer RNA with methionine. So remember, there's stems and loops. It's in the P site. The A site is empty. There's nothing there. And then we have the E site also, which is empty. So there's 3 binding sites. Typically, only 2 transfer RNAs will ever be attached to the ribosome at the same time. You won't have a transfer RNA in all 3 at the same time. Some of the animations online, some photos and images you may see, may only show 2 sites. And that's simply because that's old, based on old data, that kind of shows only 2 transfer RNAs are ever bound to the ribosome at the same time. We know that there are actually 3 sites, but only 2 are used at once. So now the next step is elongation. In elongation, what we're going to have is a new transfer RNA with a single amino acid binds to the A site. Okay, so that's going to be the first step. So we're going to get a transfer RNA. It's going to come in here. This is, say, amino acid number 2. Now we don't know yet if it's number 2. It's potentially number 2. What has to happen is that the codon and anticodon have to be checked to see if they're proper. So codon and anticodon pairing And I'll put a question mark after that, because that's kind of what's happening. It's checking, do they pair up or not? Yes or no. If yes, then we proceed forward. If no, then it goes back to the start. Then that transfer RNA is kicked out and another new one will come in. Codon, anticodon pairing, no, it gets kicked out again. New one will come in. That's the idea. So typically when the transfer RNA comes in, if it's the right transfer RNA, that means it has the right amino acid, and then we move on to the next step. So in the next step, what's going to happen is part of the large ribosomal subunit, okay, is an enzyme called Peptidyl Transferase. So this is part of the ribosome. Peptidyl transferase is going to then do a couple things. It's going to, we can think of the name of it. So transfer, talking about transfer RNA, right? Peptidyl, thinking about peptides, polypeptides, and peptide bonds. What's going to happen is it's going to break the bond between the amino acid and transfer RNA in the A site, and then it's going to create a new bond between that one and the amino acid in the A site. Okay, so breaks the peptide bond. It breaks the bond, all right, between the amino acid and transfer RNA in the P site, and then it creates a peptide bond to the amino acid in the A site. So now the transfer RNA in the P site doesn't have anything attached to it. And the one in the A site actually has 2 amino acids, so it's a small peptide. This is the point where it moves, okay? So without really being able to animate this, I'm just drawing it on this board. I'm going to show, movement in this fashion, okay? So that the ribosome is now going to move forward. And now the sites are going to change, okay? This is going to be now the E site. The site with this transfer RNA that has the 2 amino acids, that's going to be the P site. And here, now, this is the A site, and it's empty. So that means the empty transfer RNA is moved to the E site. You can see it's there. And now we're set up kind of the way we should have been when we defined, the sites of the ribosome. The A site here is for transfer RNAs with single amino acids. The P site is for transfer RNAs with peptides. And then the E site is for empty transfer RNAs that have no amino acid. This isn't going to stay there very long. It's pretty much going to get kicked out. And so you're going to then have that transfer RNA. It's not destroyed or anything. It's just removed. It's just an empty transfer RNA that has a specific anticodon, and it can then, again, in the future have another amino acid attached here, but right now it doesn't. Right now there's nothing there. So the empty transfer RNA is kicked out. It's gone. And now we'll get a new transfer RNA starting. Now we come back to the beginning. A new transfer RNA comes into the A site, amino acid number potentially 3. Everything is then just going to repeat. So we said this is repetitive. So now anticodon and codon, are they correct? If no, get out. If yes, then we're going to move on. Then this bond will break or be broken by that enzyme. Peptide bond will form. Now you have 1, 2, 3 amino acids long. They're attached to the transfer RNA that's in the A site temporarily. This one is empty, again temporarily, until the ribosome moves. Now the ribosome will move again. And see, all the sites will change. So I'll just do this one more time here, just to kind of give you the feel of it. Now there's animations and videos that show this actively, so where this is all changing, much better. It's good to kind of look at some of the static images first, or some little drawings like this, partly because some of those animations go really fast, and if you don't really know what you're looking at to start with, then it doesn't really help. But if you kind of get the basics down and then watch some of the animations, they'll make a lot more sense, and they'll be incredibly helpful for you to get kind of a picture of this as an active process. I left behind the small subunit, but it's moving along with it too. So the small subunit and the large subunit are one unit as a ribosome moving along the messenger RNA. So now again here we have the A site open, the E site has an empty transfer RNA, and the P site has a peptide. We'll just continue. The E gets kicked, the empty one gets kicked out, it gets removed. A new one will come into the A site. Bring it in here, and I'm just going to end with this. So now this is potentially amino acid number 4, and so on. And this could happen, for hundreds and hundreds of amino acids, hundreds and hundreds of codons to build this new polypeptide. It'll just keep repeating over and over and over again. Make a new peptide bond, shift everything over, bring in a new transfer RNA, make a new peptide bond, and it just continues. So that's elongation, and the polypeptide will just keep growing and growing. So now we have initiation. How does it all start? It starts with the initiation complex here, binding to the cap, finding the start codon. Then the large subunit comes in, and then we start to build. So then we bring in the transfer RNAs with single amino acids, build the peptide bonds, and then the polypeptide grows. Ultimately, though, it has to stop. It has to end. There's some ending for it. That end is then called termination. So what's going to happen, and I'm going to take a second here to erase some of this so I can write a few more things as we get into the termination phase. So what's going to happen here is fairly straightforward. We're not going to go into too much detail with termination. But it's essentially that, if you recall, there are 64 possible codons, which are the combinations of the 4 nucleotides, right? The A, U, G, and C, that could be put in groups of 3. So that comes up with 64 possible combinations. 61 codes for amino acids. So there are 3 that don't code for amino acids. They code for what are called stop codons. And so this is where we get into termination. These 3 stop codons are UAA, UAG, and UGA. Those are the 3 stop codons. So at some point in time, I don't think I just haphazardly drew one here. So I'll just, here, I'll put this one here. Now, just because in this line I have a UAG doesn't necessarily mean this would be the stop codon. It has to actually fall on it as a group of 3. Remember, I said we're breaking these into groups of 3. So essentially, as you read along here, I'll try to see how this works out. See, if we were doing it the way I was progressing, this would be a codon here. And so this UAG, even though they're next to each other, kind of forming a code, they wouldn't actually be a codon because the codons are defined by these units of 3 starting with the start codon. And you break it up into groups of 3. So it would have to fall so that there would be some other nucleotides here to change it so that it actually would fall on this. So if it falls on the UAG, then that is the stop codon. If it doesn't, then just because you see that sequence doesn't mean it's going to be a stop codon. It actually has to line up. All right. So ultimately what's going to happen is you keep moving forward. You have your polypeptide, so you have lots of amino acids here attached to the transfer RNA in the P site. So it's a very long chain. This is our polypeptide. And now we find the stop codon. So what's going to happen here, instead of a transfer RNA binding into the A site, we're actually going to get something called a release factor. So for the process of termination, we get a stop codon is red, then a release factor binds to the A site. And once the release factor binds in here, it doesn't have any amino acids with it. But that enzyme, the peptidyl transferase, is going to still try and do its job, and it's going to break the bond between the transfer RNA and this amino acid here. That's part of the polypeptide. But it can't make a new peptide bond because there's no amino acid there. So the polypeptide is released. That's what we call it, a release factor. It triggers the release of the polypeptide. Now, this polypeptide may be released directly into the cytoplasm because that's where this is happening. Or, alternatively, what we're going to get into in another lecture, is it could be pushed into the endoplasmic reticulum. The endoplasmic reticulum is a site where a lot of polypeptides are processed into final functional proteins, and they're also packaged in a way that can transport them to their final destination. So if they're going to become a membrane protein, if they're going to go back into the nucleus, depending on where they're going to end up in the cell, it's in the ER and then the Golgi apparatus where that processing occurs, and that can still happen as an additional part to this. So, but for right now, for termination of the event, we read a stop codon, release factor comes in, the bond is broken, the polypeptide is released, and then the last thing that essentially happens is that the ribosome falls apart. So the large subunit and the small subunit separate from one another, they separate from the messenger RNA, and that is the end. Okay. The messenger RNA, sometimes toward this 3' end, has some little stem and loop structures that physically acts as a sort of like a speed bump, where the ribosome can kind of hit it and then break apart because it can't really pass through that point. And so this is the end of what we call translation. And what we've done is translated a nucleotide sequence into a very specific amino acid sequence. You don't have to know or memorize in this class, the general biology class, the specific codons for the amino acids. In a biochemistry course, you may then have to actually memorize each codon and what amino acid it codes for, but we'll be using a table, and so I'll be going over the table in the next lecture, and then we'll also talk about processing of the polypeptide once this ends. Okay, and so for many proteins, that would be it.